Hey guys, my name is Didi Jimenez, and today I will be giving you a short overview of glycolysis, gluconeogenesis, and their pathways, how aldolase is involved in both glycolysis and gluconeogenesis, the different isoforms of aldolase and different expression patterns, and an examination of how aldolase B expression can be regulated by transcription factors such as HNF1, HNF3, CEB, PB, and DBP. So let's get started. We are first going to start with an ancient pathway known as glycolysis that was present in Earth's atmosphere long before there was oxygen. Breaking it down, the word glykos is Greek meaning sweet, and the word lysis means to split. In other words, glycolysis means the splitting of one glucose molecule into two molecules of pyruvate. Although this pathway was present way before oxygen, it has different effects when it is present. In the presence of oxygen, pyruvate normally enters the mitochondria, where it is oxidized to acetyl-CoA. On the other hand, when there is no oxygen present, pyruvate is reduced to lactate. In glycolysis, there are 10 reactions that take place in the cytosol, resulting in two ATP molecules that don't require molecular oxygen. Contrary to this oxidative phosphorylation that occurs in the mitochondria that generates 30 ATP molecules and does not require oxygen. Sorry, I took a drink of water. Various simple sugars have the ability to enter glycolysis like monosaccharides, glucose, galactose, and fructose. These simple sugars, for most of us, are ingested through consumption of products that contain the disaccharide sucrose, aka table sugar, or lactose, milk sugar. Sucrose is made up of one molecule of glucose and fructose, while lactose contains one or more molecules of glucose and galactose. Both of these simple sugars are broken down by digestive enzymes sucrase and lactase. So there are three key concepts of glycolysis. It is the only pathway that can generate ATP in the absence of oxygen. In other words, it is an anaerobic condition, or in cells lacking mitochondria such as red blood cells, erythrocytes. In these scenarios, pyruvate is converted into lactate. In the presence of oxygen, glycolysis generates pyruvate, which enters the TCA cycle, also called the citric acid cycle and the Krebs cycle, located within the mitochondria to produce ATP. And many of the metabolites of glycolysis and the TCA cycle can also enter anabolic pathways that generate NADPH and the building blocks needed for generation of glycogen, lipid, nucleotide, and protein synthesis. This biosynthetic pathway that glycolytic intermediates funnel into include the pentose phosphate, the hexamine, and the serine and glycerol biosynthetic pathways. Moving on to the actual steps of glycolysis. The first step of glycolysis has to do with the enzyme hexokinase phosphorylating or adding a phosphate group to a glucose in a cell's cytoplasm. Breaking it down, in this step, a phosphate group from ATP is transferred to glucose, producing glucose 6-phosphate, or G6P. During this step, one molecule of ATP is consumed. So the first step is here, our glucose molecule, and we're involving this hexokinase here, and we're getting our product, I'm going to do that in red, of glucose 6-phosphate. This is followed by step 2, which involves the enzyme phosphoglucomutase, which isomerizes 6-8P into its isomer fructose 6-phosphate, or F6P. Remember, isomers have the same molecular formula as each other, but different atomic arrangements. So in this, that's this product, I'm pretty sure. We're now working with this enzyme in step 2. As you can see the number 2, to get the product of fructose 6-phosphate, which is that chart there, the little graph there, formula. Okay. Moving on to step 3, where we have kinase phosphofructokinase, which uses another ATP molecule to transfer a phosphate group to F6P in order to form fructose 1,6-biphosphate, or FBP. At this point, 2 ATP molecules have been used so far. So next we have this enzyme, and this is our product, fructose 1,6-biphosphate. That is this chart here. And at this point, well, not yellow, we'll use a different color. Let's use purple. 2 ATP. 2 ATP molecules have been used at this point, or step 3. Okay, and the next step involves the enzyme aldolase, which is extremely important, so we'll go over it more in depth later, but in this situation, it is in charge of splitting fructose 1,6-biphosphate, which is our result from step 3, into a ketone and aldehyde molecule. The resulting sugars, dihydroxyacetone phosphate, or DHAP for short, and glyceraldehyde 3-phosphate, or GAP, are isomers of each other. Ooh. So here's our aldolase, which we get different results, 2 different results that are this one and this one. Wow, it's hard to use this marker. That one and that one. Okay. Which leads us to our 5th step, which is, it has 10 steps, so it's a lot of steps. This enzyme here, triose phosphate isomerase, which rapidly converts DHAP into GAP. These isomers can interconvert. In order to complete the next step of glycolysis, the substrate GAP is needed. Okay, so we're going to go over the next 5 steps on the next slide, because this was a lot of information. We are now in step 6 of glycolysis, which is with the enzyme glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, which is here. We'll just keep the same little star and arrow, which serves for 2 functions. In glycolysis, the first is to dehydrogenate GAP through the transfer of one of its hydrogen molecules to oxidizing agent nicotinamide adenine dinucleotide to form NADH plus a hydrogen molecule. The second function is that it adds a phosphate from the cytosol to the oxidized GAP in order to form our product of 1,3-biphosphoglycerate BPG. Our product, I think I put them in red. Our product, which is that one. Both molecules of GAP produced in the previous step undergo the process dehydrogenation and phosphorylation. Our 7th step now is with the enzyme phosphoglycerol-kinase, which is this one. I'm so sorry, I'm trying to let y'all keep up with me, but it's a little messed up. Which transfers a phosphate from BPG to a molecule of ADP to form ATP. This transfer happens to each molecule of BPG, resulting in our product of 2 3-phosphoglycerate 3-PGA molecules and 2 ATP molecules. So we get 2 of these. I haven't even been boxing our ATPs, which would be these. But that's that one. Okay, so we're now on the 8th step of this pathway, so only 3 more to go after this. At step Step 8, we can see in the picture, we have the enzyme phosphoglycermutase relocating the P of the two PGA molecules from the third to the second carbon to form two phosphoglycerate molecules. So now we're here. Glycolysis is such a long pathway, huh? We got that. Next, we see the enzyme enolase, which removes a molecule of water from 2-phosphoglycerate to form phosphophenolpyruvate, PEP, a process that occurs for each molecule of 2-PGA from the previous step. And finally, our last step of glycolysis pathway involves the enzyme pyruvate kinase transferring a P. I just went ahead and skipped this because you kind of get the point of the little enzyme here, result, and then the little graph. So we get the enzyme pyruvate kinase transferring a P from PEP to ADP. This reaction results in 2 molecules of pyruvate and 2 ATP molecules. And that would be, in red here, 2 pyruvate. And that is it for glycolysis. Well, that was a hefty pathway, but now we are going to move on to gluconeogenesis, which is a process by which glucose can be processed by the body from non-hexose precursors, specifically glycerol, lactate, pyruvate, propionate, and glucogenic amino acids. This process, in other words, is essentially reverse glycolysis. Through this pathway, glucose synthesis is facilitated by 4 enzymes by reversing 3 highly exergonic glycolytic steps, namely pyruvate, carboxylase, phosphophenolpyruvate, carboxykinase, fructose 1,6 biphosphatase, and glucose 6 phosphatase. The only thing with glucogenesis is that 3 specific enzymes are not present in all cell types, rendering it to only occur in specific tissues, primarily taking place in the liver and in the renal cortex at a lesser extent. As mentioned before, gluconeogenesis is just like glycolysis, only that it happens in reverse. However, there are 3 highly exergonic steps in glycolysis that involve the enzymes hexokinase, phosphofructokinase, and pyruvate kinase, all of which require too much energy. So in glucogenesis, this can't happen. So nature has evolved 3 enzymes that replace these. The first step in gluconeogenesis is the conversion of pyruvate to phospholinolpyruvaric acid, PEP. For this to occur, there are several steps and enzymes required. The 3 responsible enzymes for this conversion are pyruvate carboxylase, PEP carboxykinase, and malate dehydrogenase. Pyruvate carboxylase is found in the mitochondria and functions to convert pyruvate into oxaloacetate. So I want to draw a little mitochondria so we remember that. I always associate mitochondria with red. Chose to spell out the longest word, right? Mitochondria. But due to the fact that the product cannot pass through the mitochondrial membrane, it has to be converted to malate by malate dehydrogenase. Malate can then cross the mitochondrial membrane into the cytoplasm, where it is then converted back into oxaloacetate with another malate dehydrogenase. Lastly, oxaloacetate is converted into PEP via PEP carboxykinase, which are the 3 enzymes used all in one. The next several steps are exactly like glycolysis, only the process is in reverse. The second step that differs from glycolysis is the conversion of fructose 1,6 BP to fructose 6P with the use of the enzyme fructose 1,6 phosphate. The conversion of fructose 6P to glucose 6P uses the same enzyme as glycolysis, phosphoglucoisomerase. The last step that differs from glycolysis is the conversion of glucose 6P to glucose with the enzyme glucose 6-phosphatase. This enzyme is located in the endoplasmic reticulum. So we also have the endoplasmic reticulum for this pathway, specifically highlighted plasmic. It's not letting me write no more. Okay, whatever. Can I highlight this part? Remember earlier when I mentioned the enzyme aldolase in step 4 of glycolysis? Well, here we are. This protein helps to break down certain sugars to produce energy. Aldolase has 3 isoenzymes A, B, and C, and is possibly present in all cells. It occurs particularly in large quantities in the muscles, liver, and the brain. So that is extremely important because that in the muscles, because it has different functions depending on where it's at. It acts differently. In the liver, and the brain. So let's get further into it. So now let's go over aldolase involvement in glycolysis and gluconeogenesis. This occurs in the 4th step of glycolysis, where fructose 1,6-biphosphate aldolase, FBA, catalyzes the reversible conversion of the 6-carbon glycolytic enzyme fructose 1,6-biphosphate, which is on the left. Here. Into 3-carbon intermediate glyceraldehyde 3-phosphate. We go into that once it catalyzes. And dihydroxyacetone phosphate, which is, they are both on the right. So let's go ahead, 2 3-carbon intermediates. These are 3-carbon intermediates. Wow, that's hard. Okay, intermediates. The C3 and C4 carbon bond in F1CBP, 6BP, which is the one on the left, this one here, is cleaved to form GAP and DHAP, following the formation of an intermediate within the active site of the aldolase enzyme. As seen by the title of the slide, aldolase is involved in both glycolysis and glucogenesis, and even fructose metabolism. So not only these two, but we also have fructose metabolism. As I mentioned before, it is found distributed and expressed in different areas of our body. So vertebral adolase exists in three forms of isozymes, with distribution and kinetics throughout different tissues. Adolase A is in the muscle and red blood cells. Adolase B is in the liver, kidney, and small intestine. Adolase C is in the brain and neuronal tissue. The mechanisms of these adolase occur by two distinct chemical paths. Covalent catalysis is used by class 1 adolase of animals and higher plants through a Schiff-based intermediate with ketose sugar substrates, while class 2 adolase that pertains to most bacteria and fungi require a divalent metal cation as a cofactor. So this one, class 1, this would be easier if I could type. Covalent catalysis. Class 2, divalent metal cation. Cat. That's horrible. Cation. As mentioned before, among the class 1 enzymes found in mammals, there are three tissue-specific isozymes of adolase that have similar molecular masses and catalytic mechanisms. Adolase A, which we said was in muscle and red blood cells. Adolase B, which is expressed primarily in the liver, kidney, and small intestine. And adolase C, expressed primarily in the brain, smooth muscle, and neuronal tissue, which I drew those for you in the previous slides. So as an enzyme of the glycolytic pathway, adolase B's activity and mRNA levels in the liver fluctuate according to dietary status. Both the enzyme activity and the mRNA concentration decline during fasting and increase 4 to 8-fold upon refeeding of a carbohydrate-rich diet. So carbs, depending on what you eat, regulation changes. So yeah, that's kind of common sense, right? Depending on what you eat. On what you eat. That goes without saying, right? No, the saying, you are what you eat. Okay? So we have now reached the end of the presentation. These here are my references if you want to look deeper into it and further into adolase, because I just went over it quickly. But thank you guys. See you on the next one.